Here for question 4, I wanted to show you that when we are going to enter a sequence, you can either paste the sequence. So if I give you an accession number or a GI number, you can actually enter just the number. You don't need to go to the page to retrieve the sequence. So let me show you how just by entering the sequence, BLAST knows what I want. So the question is saying how many genes are predicted in this sequence here, and you're going to go through the same steps, and we're going to hope that entering just the nucleotide database, so everything, maybe we're going to get lucky and get some genes predicted. And I don't know if you noticed that when I put the number there, BLAST already found it cross-referenced, and then it knows where the sequence comes from. So the maize is corn, and let's see how many genes are predicted in that genomic sequence of corn. So we'll BLAST that, and I'm going to do the same thing on another window, and instead of all the different nucleotide sequences, I'm going to say I want you to narrow down to an RNA. Okay, well, let's look at the all nucleotide sequences. And here's how long that DNA sequence is, 68,000 nucleotides. Pretty amazing how fast it does this comparison. So let's see what it matches. And then you're like, okay, clone, whatever, chromosome 5. So this is saying some DNA sequence that came from a clone that was named this. So, and this is bacterial artificial chromosome. That's, people used to do that more in the past to sequence. We cut the DNA in big chunks and then glue those chunks to a bacterial chromosome. And that's how we made the clones. So let's see another complete CDS. Okay, oh, this has retrotransposon. So this is saying something, oh, it has some function, it encodes some gene. And these other ones are just, this is saying also from clone. So this is not actually informing much on is a gene or not. Oh, here is a gene. Let's look at the graphic summary. And then you have this, which is a lot, right? There's probably 100 rows. I don't want to go through every single row. So I could like hover over these guys, or I could look at the description, but I don't really want to do that. I only want the ones that say gene, mRNA, things like that. So instead of going through all these and narrowing down through here, then I could run, go back, instead of doing nucleotide, I can narrow it down to search only RNA. So I did this already. It's the same search. This is the same GI number of corn. Still doing BLAST N, but here I narrowed down to I only want the results that are matching RNA, and then I get a different set of results, and I get a bunch of predicted. And as I said, I want the ones that have biological information of them. I don't want the predicted. I want the ones that actually say something. So this uncharacterized, I bet this is locus, I don't know, mRNA. Okay, so there's some mRNA from this locus. Uncharacterized means they don't know what it does, but it's an mRNA, so it's a gene. It counts. Predicted, I'm skipping. So that's also mRNA, so I include. So all the ones that say predicted, I skipped. And I only marked the ones that say something that it says mRNA or protein or whatever ends in A's. There will be some enzyme. So I went through all of these and I just checked whatever does say predicted. And then the easiest way to see how many genes are predicted, then even though I selected 6, maybe there's some overlap, as in the same location in the genome is encoding those genes, and so we're going to say they're the same. So for example here, so there was a hit from corn mRNA, and then the same spot, oh, look at that, transcription elongation factor. It could be that this mRNA wasn't transcription elongation factor. So because this mRNA and this mRNA that they know the function for match this part of my query, which was my genomic sequence. So remember, this one I didn't lie. This one was genes plus non-genes. So there is a gene here, which is these two hits here. There is one gene here. So 1, 2, 3. This is a fourth gene. 5 genes. So, and then this line here connecting means that this sequence doesn't match this part, but it's this block and this block are the same hit. So if I go slowly, maybe you can read what it says. No? Okay, so this is the uncharacterized locus mRNA. It's the same one here. And it could be that this line here, the reason why it doesn't match this sequence is because there is DNA here that doesn't exist in the mRNA. So it could be that this whole chunk is an intron. And then here, lots of tiny exons and lots of introns. So the answer to this one, I count it as 5 genes because I'm looking how many genes in this, no, how many parts of my genomic sequence here contain genes. And so here there was one mRNA, and this one said it's an mRNA with known protein, but they both match the same chunk. So I'm counting this one as 1.